Hello and welcome. You're listening to Cellvita Science Talks, where we explore key challenges in drug discovery and development. In today's episode, we're discussing target engagement assays, as presented in one of our latest blog articles, A Practical Guide to Target Engagement Assays. In this article, we explore why target engagement has become a central concept in modern drug discovery, and how biochemical, cellular, and proteomic assays help researchers understand whether a compound truly interacts with its intended target. We also compare some of the most widely used target engagement technologies and discuss how they support decision-making throughout the drug discovery process. The full version of this article, including the comparison table and downloadable attachment, is available on our website. If you're watching on YouTube, you'll also see the visuals on screen. And if you're listening on Spotify, we'll guide you through them step by step. Introduction. The transition from simple assays to more physiologically relevant systems remains a major challenge in drug discovery. Compounds that appear active early on often fail to reproduce those effects in more complex biological environments. Often the underlying issue is simple. We do not know whether the molecule truly engages its intended protein target under relevant conditions. Without this knowledge, it becomes difficult to interpret efficacy, understand safety signals, or decide which compounds deserve further investment. Target engagement has therefore become a central concept in modern drug discovery. It links biochemical potency to cellular activity, and provides mechanistic confidence that a compound is acting on the right protein in the right way. Advances in biochemical, cellular, and proteomic technologies now allow engagement to be measured earlier, at higher throughput, and in more physiologically relevant contexts. In this article, we outline why target engagement matters and how different assay classes can guide better decision-making throughout a project. Why is monitoring target engagement important? Target engagement is a measure of the interaction between a drug and a target protein of interest. Although it is not always a requirement of drug regulatory approval, demonstrating target engagement and understanding the precise mechanism of action of a molecule is preferred. In the years from 2010 to 2017, 90% of drugs entering clinical trials failed to reach registration. Almost half of these failures were attributed to a lack of efficacy and target engagement, with a further 30% displaying unmanageable toxicity. It is now thought imperative to determine drug target and off-target engagement early in the drug discovery process to avoid inadequate exposure, unwanted side effects, and insufficient efficacy. What types of assays can I use to measure target engagement? There are several ways to measure the interaction of a drug with its validated target. The most appropriate assay type to choose will depend on a number of factors, such as the scale of screening, the project stage, the equipment and expertise available to you, and the types of questions being asked. Biochemical assays, readily amenable to HTS. Biochemical target engagement assays are often the first opportunity to understand whether a compound can bind directly to an isolated recombinant protein or enzyme target. Many discovery campaigns begin at this stage because biochemical and biophysical methods can be scaled efficiently, from millions of compounds in DNA-encoded libraries or highly miniaturized fluorescence and luminescence assays, down to thousands of molecules tested with biophysical techniques such as SPR, thermal shift assays, or mass spectrometry. A key advantage of biochemical assays is that they offer unbiased measurement of small molecule binding in a controlled environment. By removing cellular complexity, they enable precise assessment of parameters such as binding kinetics and residence time, both of which are valuable for prioritizing chemistry. These measurements help build meaningful structure-activity relationships, or SAR, and support the prediction of in vivo efficacy, safety, and duration of action. Because of their scalability and interpretability, biochemical assays remain a fundamental decision-making tool early in discovery. However, their results ultimately need to be complemented by cellular assays, as biochemical binding alone cannot guarantee engagement or efficacy in a biological environment. Cellular assays, measuring engagement under physiological conditions. Cellular target engagement is essential for understanding whether a compound that binds an isolated protein will do so under physiologically relevant conditions. As impactful as biochemical assays are, they do not guarantee binding or efficacy in a cellular environment. An appropriate functional response in a disease-relevant cellular background is also not, on its own, sufficient evidence of target engagement. For this reason, dedicated cellular TE assays provide an important complementary perspective. Several high and medium throughput cellular assays are now available and can be deployed in disease-relevant cell backgrounds. Although these assays were historically used later in the drug discovery cascade, there is now the possibility to screen compound engagement at scale during hit identification. Many of these systems rely on miniaturized, simple workflows that measure engagement through sensitive fluorescent or bioluminescent readouts, making them highly amenable to automation and higher throughput formats. By assessing target engagement in a cellular context, these assays help determine whether compounds reach and bind the protein of interest in the presence of native biomolecules, under conditions that more closely reflect a biological system. This information provides crucial confidence when interpreting biochemical hits and selecting compounds for downstream optimization. Proteomics, mapping on- and off-target engagement at scale. Proteomic approaches add another dimension to target engagement by enabling the detection of both intended and unintended interactions across the cellular proteome. In addition to measuring binding to the primary target, many of the same technologies can be used to assess off-target interactions that may contribute to toxicity or unexpected biology. This broader view is increasingly important for de-risking candidates before they progress into advanced studies. Off-target engagement can be measured using techniques such as tandem mass tag or data-independent acquisition mass spectrometry, as well as chemoproteomic analyses that identify small molecule protein targets using chemical probes and MS. These methods allow systematic evaluation of protein engagement across large sets of cellular proteins and can highlight unintended interactions that may otherwise remain undetected. By understanding how a potential therapeutic interacts with proteins beyond the primary target, proteomic assays support early identification of liabilities and help guide chemistry towards more selective molecules. This makes proteomics a powerful complement to biochemical and cellular assays, particularly when assessing safety, specificity, and mechanistic hypotheses. Representative target engagement technologies. To illustrate how these assay classes translate into practice, we highlight a small set of widely used target engagement technologies at Cellvita. These represent different assay principles and project stages. For a more comprehensive overview, you can find a downloadable attachment on Cellvita's website. Surface plasmon resonance, or SPR, is a medium-scale assay that uses direct measurement of Kd via mass change on binding of a molecule to an immobilized protein, as well as ternary complex formation. Its key advantages are that it is highly sensitive, label-free, and unbiased. It also allows kinetic measurements, residence time measurements, and cooperativity measurements. One limitation is that protein coupling can alter protein conformation. TR-FRET and NanoBRET are medium-scale cellular assays that measure loss of luminescence when unlabeled ligands displace a labeled tracer from a known binding pocket. They are used to assess competition binding and ternary complex formation. One of their main advantages is that they can be performed both as cell-based and biochemical assays. In cells, they measure physiologically relevant target engagement because cellular biomolecules are present and the target protein remains in its native state. These assays do not require protein purification, can measure endogenously expressed protein, are plate-based, and imaging readouts can provide additional high-content cellular measurements. Their limitations are that compounds must be cell-permeable, and both the target protein and tracer must be tagged. The Chemical Protein Stability Assay, or CPSA, is a medium to high-scale assay, similar in principle to marketed thermal stability assays. After ligand binding, the protein target is treated with a chemical denaturing agent, and the proportion of the protein in its folded and denatured states is assessed. On ligand binding, the protein becomes more stable with higher tolerability of a chemical denaturant, resulting in a shift in the denaturant concentration response curve. The assay measures direct binding. Its advantages include being cell lysate-based, high throughput, with no requirement for high-temperature incubations, lysis steps, multiple assay plates, or transfers. It is also cost-effective, provides comparable data between chemical and thermal denaturation technologies, is unbiased and sensitive, and multiple detection methods have been validated. Capillary-based Western blot measurement allows both the ligand and protein to remain unlabeled using antibody detection, while the assay can also be measured using alpha-screen, high-bit tagging of endogenous proteins of interest, or mass spectrometry. Its limitations are that capillary-based Western blot measurement has lower throughput, and the assay is not yet compatible with intact cells. Data-independent acquisition mass spectrometry, or DIA-MS, is a medium-scale assay that measures all peptides in a sample to determine binding partners. Cellular proteins are fragmented and analyzed directly by LC-MS, and proteins are identified by matching them to a spectral library. The assay measures direct binding. Its advantages include being a label-free approach, the ability to run at scale, reliable measurement of proteins, including those of low abundance, and high sensitivity. Its limitations are that it requires complex data processing and a high-quality spectral library. Conclusions. Target engagement is no longer a downstream confirmation step, but a central part of early decision-making in drug discovery. By combining biochemical, cellular, and proteomic approaches, teams can determine not only whether a compound reaches its intended target, but also how selectively and under what conditions it does so. This information helps explain biological outcomes, supports medicinal chemistry optimization, and reduces the risk of advancing compounds with unclear mechanisms of action. As new technologies continue to expand the range and sensitivity of target engagement assays, integrating these measurements early and consistently offers a clearer path from initial hits to confident, mechanism-driven candidates.